Good morning, everyone. My name is Gina Lopez, and today I will present the main findings of my PhD research funded by PhenoRob. My thesis is entitled Field-Based Evidence of Root Plasticity Under Abiotic Stresses on Tillage. Why focus on roots? Around 95% of global food production still depends on soil. However, land degradation, including erosion and nutrient depletion, is accelerating. According to the IPCC, converting natural ecosystem to conventional agriculture can result in 20 to 60% losses of soil organic carbon. Roots are central to nutrient and water acquisition, carbon input, soil, and structural stability. They are therefore essential for resilience against drought and nutrient stress. If soils degrade, roots weaken, and weaker roots accelerate degradation. It becomes a reinforcing cycle. But before moving forward, let me briefly explain some terms that I will use along my presentation. The first one is the total root length, that is here, that is the sum of all root segments in centimeters normally. The root length density, that is the root length per soil volume. The total root biomass, that is the dry mass of all roots. The root mass density, that is the biomass of the roots, per soil volume. The specific root length, that is the root length divided by the root biomass. The rooting depth, that is the maximum depth that the roots can achieve. And there are some ratios that involve above, below ground. One of them is the root to shoot ratio, that is the root biomass divided by the shoot biomass, and the root length per shoot biomass. Also, I will talk about root plasticity, which refers to the ability of roots to adjust their structure in their response to the environmental changes. Studying roots presents a methodological challenge. Laboratory and greenhouse methods provide a strong control, but often produce unrealistic root systems. Field experiments, otherwise, reflects agronomical reality, but are variable and labor-intense. My research prioritizes field reality and connects it with soil crop modeling because it's well known that better representation of roots in models will result in better predictions of yield and carbon allocation. The general objective of my work was to quantify how nutrient stress, tillage, and climate pressures reshape the root traits and the root to shoot ratio. This was addressed through 4 research questions. The first one is to investigate a global root responses to nitrogen, phosphorus, and potassium deficiency. The second was investigating root responses to nutrient deficiency in a long-term field experiment. The third one was to investigate the effect of tillage on root allocation, and the fourth one was the evaluation of soil crop models under these climate drivers. Here for research question 3, across 28 studies, we found that switching between conventional and no-till system had minimal impact on the root to shoot ratio. Therefore, I will not discuss this in detail today. So I will talk more about the other research questions. Let us start with the first research question. This is how do root traits, particularly root length, root biomass, specific root length, and the root to shoot ratio, change under NP or K deficiency across major arable crops at field scale. To answer this question, I made a study entitled Nutrient Deficiency Effects on Root Architecture and Root to Shoot Ratio in Arable Crops that was published in Frontiers in Plant Science and has already more than 200 citations. For this study, I conducted a systematic review across major databases, focusing exclusively on field experiments. We excluded POT and laboratory studies to maintain field relevance. After screening, 50 peer-reviewed field studies were included. Only studies reporting quantitative root traits were analyzed. And to ensure a comparability across the environments and soil types, we applied min-max normalization and relative change analysis. Let's start with the results. Here we see in this figure shows normalized responses on the nitrogen deficiency. On the y-axis, we have the normalized values. On the x-axis, we have the different root rates. Gray boxes represent deficiency conditions, and white boxes represent the non-deficiency condition. 1 star is, stands for significant differences at 0.9, and 2 stars represent the significant differences at 0.95. Also, we have the NP that stands for number of publications, and NR that is the number of observations. Under nitrogen stress, we can see that the root length decreased by about 9%, and the root biomass also has a significant decrease by 7%. The root to shoot ratio has an increase of 44%, and the root length per shoot biomass had an increase by 33%. This demonstrates adaptive allocation. The plants invest relatively more in roots when nitrogen is scarce. Let's continue with the root plasticity under P and K stress. Under phosphorus deficiency, we have similar graph than before, but we can see here that under phosphorus deficiency, we have a decrease in the root length by 14%. And the root biomass also had a decrease by about 25%. And the root to shoot ratio also had an increase, but in this time was not significantly different from the both treatments. What we found is the roots became shallower with denser root hairs. For potassium deficiency, we found only 6 studies, and the trend suggests a reductions in root length and biomass, but results were not statistically significant. So the key conclusion from my research question 1 is that nitrogen deficiency is the dominant driver of root plasticity, and definitely root to shoot ratio is dynamic. It's a dynamic rate that changes with the stresses. However, responses depend to many factors. For example, crop species, the stress severity, soil conditions, climate, and management. But this study provides us with a baseline for the field experiment. So moving forward, I wanted to conduct my own field experiment to address some gaps identified in the global review. And in this study, I wanted to answer the question in a long-term field experiment, how does the sustained omission of nitrogen, phosphorus, potassium, or lime alter seasonal winter wheat shoot and root development and the root to shoot ratio? For that, we published an study. Studies winter wheat shoot and root phenotypic plasticity under fertilized and nutrient-deficient field conditions. For this, we conducted this field study at the long-term fertilizer experiment in Dikov South, that is near to Cologne. That experiment was established in 1904 and has a lowest loam soil. The mean temperature is 10 degrees, and the annual rainfall is 750 millimeters. The study crop was winter wheat during the growing period of 2019-2020 and 2020-2021. 7 treatments were studied or tested: fully fertilizer treatment with manure and without manure, and omission treatments, omission of N, P, K, and lime omission, and one treatment without any fertilizer. We sample soil, shoots, roots, and especially for roots, we sample down to 1 meter depth across different key growing stages, as we can see here: BBCH 23, BBCH 43, and BBCH 16.9. The roots after sample were washed, scanned, and used, and we use wind riser to analyze for the architectural trace. On this same slide, we see the box plots of the total root length of winter wheat measured to a soil depth of 1 meter. And the x-axis shows the different nutrient treatments, including the fertilizer and the nutrient omission treatments and the unfertilized treatment. The y-axis represents the total root length in centimeters. And the different panels are the three growth stages. BBCH 23, that is tillering, BBCH 43, that is stem elongation, and BBCH 69, that is flowering. Here you can see the dashed red line in each panel. That is the median of the fully fertilized without manure treatment. This is as base to compare with the other treatments and to see if there are some differences with the other treatments. We can see here that at the beginning of the growing period, the fully fertilized treatments tended to maintain relatively high root length, while other treatments had relative lower root length. We can see here, right? Except in the lime omission that we have similar length. However, later in the BBCH 43 and BBCH 69, PK and lime omission treatment show recover and an even greater root length compared with the fully fertilized treatment. You can see here higher. However, yeah, the nutrient, the nitrogen omission and the unfertilized treatments show reduced root length in all stages. We can see here in the next slide the root length density across different depths. Here, the x-axis shows the root length density, and the y-axis shows the soil depth. As is well known that the root density decreases with depth, as expected. Yes, we have more in the topsoil. The fully fertilized treatment with manure and the P and K omission treatment show a greater rooting across different profiles. We can see here, and the same. And then the nitrogen omission, that's the blue, dark blue, and also the unfertilized treatment, the red, always had consistently the lower root density. On this slide, we see the observed mean total root biomass across treatments and growth stages. This graph has the same elements as the root length graph we have already seen. Looking at the first growing period in BBCH 23, the trend is similar to the root length. So yeah, fully fertilized and then lower in the omission treatments. However, during the BBCH 43, the fully fertilized treatment has a biomass similar to the PK and lime omission treatments. If you remember, the root length of that omission treatments was longer than the fully fertilized treatment. But this was not the case for the root biomass, highlighting that the root plasticity is not the same across all traits. Sometimes is greater and sometimes not. In the growing period 2, at BBCH 69, we see a greater biomass in the omission treatments here, except in the N omission and unfertilized treatment. In the N omission, because the unfertilized treatment, as you can see here, is similar, the biomass, to the fully fertilized treatment. Additionally, when we see the root diametral distribution at early growth stages, the nitrogen omission and unfertilized treatment exhibit a more than 50% very fine roots compared to about 35% in the other treatments. Overall, this indicates that nutrient stress not only reduces total length or biomass, but also shifts root morphology towards more finer roots, reflecting some adaptivity plasticity under limitation. On this slide, we see the observed dry meta-grain yield across treatments for the 2 growing periods. And the x-axis shows the different treatments, and the y-axis is the yield in tons per hectare. And the red lines, dashed line indicates the median of the fully fertilized treatment as before. Overall, the grain yield was higher in growing period 2 than compared with growing period, was higher in growing period 1 that compared to growing period 2. But the straw yield follow a similar trend between treatments and remain relatively stable across years. The clearest pattern here is that nitrogen omission and the unfertilized treatment reduce the grain yield in both growing periods. And in contrast, phosphorus, potassium, and lime omission had comparatively minor effects under the soil, so the, the, the yield was comparable. While roots show plastic responses under nutrient stress, nitrogen deficiency ultimately constrains both below and above ground productivity. So yeah, answering our second research question, uh, we can conclude that sustained nutrient omission reduces shoot biomass but increased relative root investment. Nitrogen deficiency was definitely the strongest overall impact. And phosphorus shows early reduction in the root traits, but later with partial recovery in the later stages during the growing period. And also we found that potassium effects were mild in these soils. And yeah, that was all. With research question 4, we move from field observations to model performance. The question here is how accurately do widely used soil crop models simulate root biomass, shoot biomass, and especially the root to shoot ratio? And how sensitive are they to key environmental drivers such as nitrogen deficiency, drought, elevated CO2, warming, and tillage? This is important because models are increasingly used to predict yield stability and soil carbon sequestration under climate change. But these predictions depend strongly on how carbon allocation between roots and shoots is represented. So the central issue is not only whether models simulate biomass correctly, but whether they capture the plastic root to shoot adjustment that we observe in the field. To do this, we publish a paper, and it's called The Overlook Effects and Environmental Impacts on Root to Shoot Ratio in Experiments and Soil Crop Models. And to evaluate the model performance, we use a two-part approach. The first was we relied on the meta-analysis and long-term field experiments to define the observed root to shoot responses and the different environmental drivers. And the second one was we ran a simulation using widely applied soil crop models like Monica, Sticks, Lintul 5/Slim that we use it in a platform called Simplace, and AgroC. All models were calibrated using data from Campus Klein-Altendorf winter wheat experiment from 2020-2021. We include leaf area index, shoot biomass, yield, and root biomass that we sample down to 1 meter depth. And the objective was to test whether these models can reproduce the magnitude and direction of the observed root to shoot responses under different environmental stresses. In this slide, the scenario design is shown. One factor, you can see here the factor we tested. One factor was varied at a time. So we have CO2 concentration, temperature, precipitation, nitrogen availability, and tillage that was at the end, the bulk density changes. No? Each scenario includes a reduced scenario, a baseline scenario, that is the current conditions, and the augmented scenario. This enhancing the different parameters. We vary one factor at each time, and this is isolate model sensitivity to each driver. Looking at the results for the benchmark, the data from the literature review is shown in this table. The data is summarized for common crops and specifically for winter wheat, since this was our reference crop, and then we model also this crop. The nitrogen deficiency increased the root-to-shoot ratio until 64%, and also drop increased the ratio, the root-to-shoot ratio. Elevated CO2 had a modest positive effect, and warming had a negative effect, reduced the root-to-shoot ratio. And tillage had almost no impact on the root-to-shoot ratio. As I said, that was the data that we use as benchmark to compare later with our model results. In this slide, sorry, the figures show the simulated root-to-shoot ratio under baseline conditions for our four models that are here. On the x-axis, we have the time from crop emergency until harvest in August, and in the right panel, from March 1st to until harvest. And the y-axis represents the root-to-shoot ratio that is simulated. The black X that you can see here is the observed value from the field experiment, while the color lines show the simulated daily root-to-shoot dynamics for each model. Looking at the full season at the left panel, we see clearly differences in early growth stages. The models diverge strongly in both magnitude and shape of the root-to-shoot curve before March. In contrast, when we focus on the period from March onwards, that is shown in the right panel, the simulation converge more closely and fall within the observed range, let's say. That is for the baseline, and in this slide is this slide shows the simulated root-to-shoot ratio under the different stress scenarios. Each panel represents one environmental driver, such as nitrogen deficient, drought, temperature changes, and the x-axis is are all the models and the representations, and the y-axis represents the percentage change in the root-to-shoot ratio relative with the baseline scenario. Each color box is one of the four models. When we look across the panels, we can see that the model responses differ strongly in several cases here. In several cases, the boxes are close to zero, indicating that little or no sensitivity of the root-to-shoot ratio to the applied stress. However, the sensitivity of either shoot or root biomass itself is different. Sometimes both biomasses or root-to-shoot are sensitive, and therefore the effect of root-to-shoot gets canceled out. For example, in the nutrient deficiency scenario here, sticks, that is the purple one here, showed a highly sensitivity in root and shoot biomass. But since both were in the same direction, the relative change in the root-to-shoot ratio is almost null. Temperature was the most unstable factor. Only Monica showed 150% changes in relative change in root-to-shoot in colder conditions. The others show relative changes close to zero. However, if we look at the model relative change in root biomass in temperature scenarios, the graph that we have below, we can see that the sensitivity from all models is huge. If you can see here, in colder scenarios, the relative change ranges from minus 20% to minus 100%, and in warmer conditions, especially agro-C and Simplace were also very sensitive. So yeah, it all depends from the model. Overall, the graphs show that the model sensitivity depends heavily on how carbon allocation rules are implemented. Many responses are weak, inconsistent, or do not match the direct observed in-field data. Okay, to answer our research question 4, we know that most models lack mechanistic stress-responsive allocation rules. CO2 effects are underestimated, while nitrogen responses is captured only partially by MONICA. Drought responses are weak or inconsistent in another direction. And therefore, we can conclude that the current soil crop models do not adequately represent root-to-shoot plasticity. And that brings me here to the agronomical implications of this root plasticity. This work demonstrates that roots are not passive structures. They respond dynamically to the environmental stresses. Across field studies and long-term experiments, we see that nutrient limitation, especially nitrogen deficiency, significantly alters a root architecture and root-to-shoot allocation. However, increased relative root investment does not necessarily compensate for the reduced shoot growth. In fact, nitrogen remains the dominant constraint on overall production, so N deficiency crops require a target and strategies. P is also critical since it's rapidly lost due to erosion. For that reason, we need P-efficient system with fine root foraging cultivars. Also, from my findings, we know that key strategies should prioritize physiological tolerance rather than root-based adaptation, since we didn't find too much effects on the roots. Looking forward, root plasticity has more important implications for climate resilience agriculture. And their draw and increased CO2, moderate end stress can increase root allocation, enhancing water and nutrient acquisition. However, our result also shows that in a strong nitrogen deficiency, total biomass declines substantially, even in root-to-shoot ratio, even if the root-to-shoot ratio increases. Warming is a factor that reduces the root investment, and it worsening the drought tolerance, nutrient uptake, and soil ensuring. I can also conclude that minimum tillage systems or not tillage systems not only have zero consequences in root-to-shoot ratio, but also protect the soil structure. We have here that the stress responsive roots help maintain yields, both soil carbon, and reduce degradation risk. This suggests that breeding strategies targeting adaptive root traits, such as deeper rooting or finer roots, could improve resilience. Finally, future work could focus on going deeper into some gaps, such as the effect of K deficiency and raw, CO2 enhancement and warming on roots and root-to-shoot ratio at field scale. Could be another area of study. We put a special focus in field scale because these increase the agronomic relevance. Also, the next step is improving how models represent root processes. Our findings show that current soil crop models often lack a mechanism allocation rules for stress-driven root plasticity. Future work should focus also on integrating dynamic carbon allocation and explicitly respond to the different stresses. Additionally, more high-resolution field data on root distribution and seasonal dynamics are needed to validate these models, and improving this representation is critical for more reliable prediction. And yeah, I would like to thank my supervisors. I would like to for their guidance. I would like to thank also PhenoRob project for funding my research, and the colleagues in Crop Science Group at INRES for discussions and collaborations. And thank you all of you for this for your attention. And I am happy to take questions.